Hi students, let's talk about mutations, and now the different type of mutations among the bacteria could result in the formation of certain biochemical mutants. What are these biochemical mutants? As a result of certain mutations or sudden changes, the biochemical pattern or the biochemistry of the cell would get changed. What is the biochemistry of a cell? Usually, there are certain biosynthetic pathways which are there for the synthesis of various amino acids as well as nucleotides, etc., within a microbe as well as within a body, a microbe as well as an organism also. Sometimes, because of the action of mutations, these biosynthetic pathways would be mutated, and this would result in the formation or the elimination of the capacity of the mutant to make that particular essential molecule, let it be an amino acid or a nucleotide. So when such mutations happen, they are referred to as a biochemical mutants. And when such a mutation happens, the strain which bears such a mutation has a conditional phenotype. That is, it will be unable to grow in a media lacking that molecule, but grows grows when the molecule has been provided. And such mutants are called the auxotrophs, and they are said to be auxotropic for the molecule they cannot synthesize. Now, I'll give you an example. If you have a microbe, it was first capable of growing in a media even with it is devoid of methionine. Methionine is an amino acid. It can synthesize its own methionine. So, even if the media does not contain methionine, it will grow. That was a wild type of strain. Okay, and consider we'd call them as prototrophs. The prototrophs are those which are able to grow in a minimal media containing only salts. They don't need various other things and all. They can synthesize. Even if you give the common salts like nitrogen, phosphorus as well as a carbon source, they can utilize these elements and synthesize their own amino acids. That was a strain. That was the first strain that we have, and we are calling it as prototroph. Now, if this prototroph is undergoing some UV exposure or any other mutagenic factor, any mutagen exposure, it will result in the formation of another strain which is not capable of synthesizing methionine. That is, the methionine biosynthetic pathway of that particular organism is being interrupted or being disrupted, or it is being mutated by the exposure of that mutation causing agent. So in such cases, the organism cannot grow if methionine is not being provided. If methionine was an essential substance for its growth, it will not be able to grow unless and until methionine is being given in the media. And such a mutated organism is referred to as your auxotroph. So let's just go into an example of here. This is an arginine auxotroph which has been given. In a minimal media, you refer to as a prototroph. I told you earlier, it is an organism which can grow in a minimal media. What is a minimal media? A minimal media will have a carbon source, a nitrogen source, as well as various other phosphorus source and all that, because for the synthesis of DNA as well as for amino acids, we have certain basic elements. So if you have an organism here which is capable of growing in a minimal media, or in this media there is only carbon, nitrogen, phosphorus is there, and all other essential amino acids are not there. Only the building blocks of all these are there. If the organism is not mutated, it will utilize these minimal media minerals, and it will grow. And here this is another media. So, but a auxotroph will not be able to grow. We are looking this is an arginine auxotroph. So it requires arginine to grow. Without arginine, it cannot grow in this minimal media because it does not know how to synthesize arginine from using the components of this minimal media. And here there is another minimal media with arginine, provided with minimal media with arginine also. This is growing here. A prototroph minimal media with lysine, that is also, it is growing here. And Here I am adding minimal media with arginine and it is capable of growing here, and I am putting here minimal media plus lysine. Even then this is not able to grow because it does not have arginine. So an auxotroph is something which has been found to be mutated in its biosynthetic pathway. Okay, it has been mutated and hence it cannot grow in the absence of a preformed essential amino acid or a different or during for the nucleotide synthesis. Now since we are talking about growth here, we are talking about the amino acid synthesis. Okay, so either it's the biosynthetic pathway which has been used for the synthesis of a particular amino acid or for the synthesis of a particular nucleic acid has been interrupted by the action of mutagens. Such mutations are referred to as a biochemical mutations. And the wild type of strain which is capable of growing on the minimal media in the presence of nitrogen, phosphorus and a carbon source is referred to as a prototroph. Well, the other one which gets mutated is referred to as a auxotroph. And another type of biochemical mutant which we come across is a resistant mutant. Now these mutants, for example, they acquire resistance to some pathogens, chemicals or antibiotics. Now you might have come across the MDR strains, okay, the multi-drug resistant strains, MRSA like methicillin resistant Staph aureus. These organisms are getting what? They are becoming methicillin resistant. So it is a mutant. If the wild type of strain of Staph aureus was not resistant to methicillin, and if it undergoes a mutation and it gains the resistance against that particular antibiotic, in our case methicillin as we mentioned here, that is a resistant mutant. And auxotropic as well as resistant mutants, they are quite important in the microbial genetics due to the ease of their detection and their relative abundance. Now here this principle of auxotropy and prototropy has been used to identify various mutants. Now for example, when you do the process of genetic engineering and all that, the identification of clones can be done by either auxotropic based methods or by antibiotic resistant based isolation methods. You will be understanding that once we go through the steps of the genetic engineering and how we select the mutants from the vector borne mutants and all that. So with this I hope this is clear to you. With this we come to an end of the session. Thank you.